Let me kick this off now. As Bill pointed out, I'm being the moderator to the moderator, but I will hand that off to him.
We have another moderator too.
And there's another, okay. Anyway, I'm going to introduce Bill up. He will be handling this next session. Bill is the founder and principal of W.J. Pignatto and Associates, a Boston-based consulting firm specializing in global regulatory affairs strategy focused on in vitro diagnostics, medical devices, companion diagnostics, precision medicine, drugs, diagnostic co-development, and the regulatory deployment of new technologies. Previously, he was vice president, global head of regulatory affairs, companion diagnostics, where he led the companion diagnostic regulatory program at Novartis. He worked at Genentech, leading the regulatory efforts specific to the company's companion diagnostics initiative, brings over 30 years of regulatory experience, worked on more than 50 drug diagnostics co-development programs, and he has held several other positions I won't go into right now. He's been active in numerous FDA industry cooperative activities regarding regulatory policy impacting diagnostic devices, spoken widely on regulatory issues, and associated with commercialization of in vitro diagnostic technologies. And with that, I will turn the floor over to him.
Thanks, Tom. Can everyone hear me okay? Okay. Nice to see some old friends and colleagues and some people that I used to work with. I like to say in the diagnostic world, you can't hide from your past. Hopefully you have a good one. We have a good session today on current issues and regulatory affairs, and given what's happened over the last couple of weeks, what was current this morning may be old news this afternoon. But we're going to be talking on regulatory issues. There'll be a panel discussion with a representative from FDA who's also going to be on that panel discussion. And there was a lot of emails going back and forth with the panel the last few days regarding the MSK and Foundation One clearance and approvals, and so that'll be an interesting discussion. I'm going to just touch on partnering and be a person talking about precision medicine, where we go from here. So our first speaker is Jeff Allard. I know Jeff for over 20 years, and we've worked together on certain projects. And Jeff is, his background, he's a biochemist, and he's current president of Lakeside Life Sciences, and provides consulting services in biospecimens, on biospecimens to the biotechnology industry. He served most recently as the director of Caris Life Sciences. He was, prior to that, vice president and chief scientific officer at Fuji Radio, and he was involved, I think it was the first IVD HDE approval. There have been some that have followed since that time, including a companion diagnostic. Prior to that, Jeff held a position of Vice President Clinical Research and Development at Immunicon, involved with circulating tumor cells. And where I know Jeff is his work at clinical and regulatory oncology at Bayer Diagnostic, and Jeff is going to speak about the regulatory considerations on US FDA submissions for a companion diagnostic test. Jeff?
Good. Is that on? Okay. There we go. Thank you. Yeah, the opening slide is, this is where we live. And Steve, if there's nothing on the map for North Dakota, there's nothing on the map for New Hampshire either. We live in the Lakes region of New Hampshire, and this is pretty much the view that we see every day driving into work, and thus the name of our company, Lakeside Life Science. I'm going to give a very pragmatic introduction to the regulatory features around IVDs. There are going to be a number of graduate level classes following my talk. So I thought I'd start you off with some of the basics, and I noticed that Michelle talked about the research and discovery process and thinking outside the box, and then we had a wonderful presentation on development of drugs, and then Steve presented development of tests and what happens when they're commercialized. But what I noticed is nobody talked about the regulatory piece. And I think it's a lot like that cliche cartoon that you've all seen of the physicist at the blackboard and he's doing the incredibly complex equation, you know, x cubed to the 12th divided by the square root of theta, blah, blah, blah. Then a miracle occurs, and then he keeps going with his equation. I think regulatory bears a lot of resemblance to that, where people just don't see it very clearly. I'm a researcher by training. I'm not really a regulatory person. I was thrust into that role due to a vacuum in our company at the time. This was prior to when Bill became a part of our company, and nobody had PMA regulatory experience. I was doing research, and research was, the funding was drying up rapidly in those days, so we moved into clinical trials and doing studies and submitting them to FDA. So my training's very pragmatic, hands-on. You won't see a lot of fancy definitions here, but I hope I'll give you some introduction and some background to terminology that we use in this industry and how it relates to the regulatory process and working successfully with FDA. Every day I get people asking me, what does FDA want? And I'll answer that question as we go along. Oh, I was going backward. Okay. So, but I will start with one formal definition. This is FDA's definition of a companion diagnostic, and it's pretty obvious. It's a good definition. In vitro diagnostic device that provides information essential for the safe and effective use of a corresponding therapeutic product. So one minor distinction that I'll point out in the regulatory realm, in the pharma world, you demonstrate safety and efficacy. In the diagnostics world, we demonstrate safety and effectiveness. Don't ask me why. I don't know that anyone knows why, but that's the terminology that we use. So just some definitions. I'd like to start with analytical testing, because this is often confusing to people. It's often called analytical validation, and I was lectured in a letter from FDA about a year or so ago, because I used the term analytical verification, and they didn't like that. But I believe it's the more appropriate term, and the reason, two reasons. One is that we use the term validation repeatedly in the things that we do. There's analytical validation, clinical validation, software validation, and we do other validations. And I think it gets confusing. So if we can separate this out just a little bit, I think it's helpful to people in our industry. But in addition, if you look at the origin of the term, you're verifying that the assay meets its specifications. The reason we do this testing is to show that we have sat down and we thought about this in the beginning. The process should be you develop this test. There's a corporate decision to pursue the test and commercialize it. R&D now sits together with marketing, and you come up with a list of specifications for what the test has to do in order to be scientifically, medically, and commercially successful. That's where you get your list of goals. And some companies call it goal specifications. Some call it the TPP, target product profile. This is a common terminology. Doesn't matter what you call it, but you need a list of specifications. And when you verify the assay, you demonstrate that the assay meets those specifications. When you validate the assay, you demonstrate that it's fit for its intended use. That's the clinical demonstration, that the test works clinically. And then there's been, for many years, this distinction. There are different, laser's not working. Anyway, wrong direction, haha. There's been for a long time this distinction between clinical validity and clinical utility. And it's important to understand in our field because this is the FDA bar, clinical validity, not clinical utility. Clinical validity means there's an association with disease. And companion diagnostics don't give a good example here because companion diagnostics are typically demonstrating clinical utility. Our test points toward the population of patients that will respond to this disease. Well, that's an outcome, and therefore you're demonstrating clinical utility. But let's use PSA as an example. Everybody's familiar with that. The PSA test, as PSA values rise, the probability of finding prostate cancer on a biopsy rises in parallel with that. So there's an association with disease. That's good enough to go through the FDA with PSA. That's clinical validity. Clinical utility shows clinical outcomes. Why do this? Why discover prostate cancer in a man? And of course that today is very controversial. Should you discover prostate cancer in men? But that aside, the question becomes, are you doing the man a favor? by finding prostate cancer? Is he going to live longer? Is his health care going to cost less money? In the cardiovascular world, congestive heart failure, we look at things like rehospitalization within 30 days. That would be an outcome study, and that would represent clinical utility. Payers often want to see clinical utility, but FDA does not require it. FDA requires clinical validity. So it's an important distinction. Predictive versus prognostic, again, it's important because it has regulatory implications. Prognosis is the more general outcomes. A good example is when I worked on circulating tumor cells, we demonstrated that if there are any residual cells detectable in blood, it is unlikely that a patient that is currently on therapy will benefit from that therapy. And we used overall survival and progression-free survival to demonstrate that. Well, if you do that, that's a general outcome, and that's prognostic. Predictive, of course, leads us right to companion diagnostics. Predictive tests predict the response to a specific therapy. Prognostic is more general. Predictive is very specific. HER2/neu testing using FISH or using immunohistochemistry predicts response to Herceptin therapy. That's a predictive test. And the importance is that these fall under very different regulatory categories, and I'll show you the categories in just a moment. But prognostic tests can be a class 2, de novo, if it's a new to the world test, and predictive would become a PMA, a class 3. So it's not just a scientific or intellectual distinction, it's actually a regulatory distinction as well. And then we've got this complementary versus companion diagnostics. And this is a very important distinction, and I hope to explore this further in our panel discussion. I have some skepticisms myself about complementary diagnostics. But clearly, companion diagnostics are predictive. They point toward patients that may or may not respond to specific therapies. Complementary diagnostics would be an instance where you run the test and it gives you information, but it is not required in order to place the patient on therapy, nor does it exclude a patient population from being placed on therapy. It's helpful, but it's not required, and you don't have to have a positive test result in order to place a patient on therapy. That raises a lot of questions in my mind, and hopefully we can talk about that in our panels. And then just another quick issue, we look at things very similar actually to the drug world in terms of interventional versus observational, but the work that we do clinically winds up being very different from what you do in the pharma world. 99% of our studies are observational. Back to the PSA example, we draw blood from a patient, they get a biopsy, we find out what the result of the biopsy was. We haven't influenced patient care in any way. All we did was draw blood, and the patient was coming in for a biopsy anyway. So we didn't mandate any medical procedures, we didn't do anything invasive, we don't place things in people's bodies, drugs, stents, whatever. So our studies are almost always observational. The one setting where our studies can become interventional is when the result of the test is used for patient care. So, excuse me. So if you're actually doing a companion diagnostic drug study and you're using the test to place the patient onto the treatment, that now would, of course, be an interventional study. And on the device side, we would need what's called an IDE, in vitro device exemption. On the drug side, IND. For us, it's an IDE. But 99% of our studies are exempt from the exemption, if that makes sense to you. But we are exempt from the exemption. And it really falls on, it's determined by, there is a guidance document from the FDA that defines significant risk studies or non-significant risk studies. And it's quite straightforward and understandable. And by the way, the determination is made by the sponsor. In our case, we don't have to go to FDA and ask, are we a significant risk or a non-significant risk? But if you're not sure, and I have had an example just within the last year where I did submit to the FDA because I just wasn't sure. I thought we were a non-significant risk study, but I wasn't positive of that. So we did go to FDA, and they came back and said, no, you're not a significant risk, you're a non-significant risk study. We were right, but I wasn't positive of that. So you can ask, but you don't have to. So how are these regulated? Most of you are probably well aware of this, and I apologize if you are, but there's a couple of distinctions here that relate to companion diagnostics that you might find of interest and value. There are 3 classes of tests. We don't just submit PMA, BLA, as you do on the drug side. Class 1, we just notify the FDA. And a common reagent that's a class 1 would be immunohistochemistry, an antibody. It binds to target X, and pathologists use it in the lab to help them to identify tissues, targets, whatever. Those are typically class 1 devices, and they are exempt from a submission. Now, everything I'm going to say has exceptions, so none of this is 100%. There are class 1 tests where you have to submit a 510K. But for the most part, they're exempt. But what you do, it's very much like the European system, CE marking, if you're familiar with that. You do have to follow GMP, you have to register with the FDA, and you have to notify them that you're selling this device, but you don't have to submit, wait, get approval before you sell the device. Class 2 today has 2 options. This system was designed in 1976, and it rapidly became obsolete. And FDA wanted to move to a risk-based system, not just class 1, 2, and 3. So they did that, and in order to develop an option for a moderate risk new device that didn't force it to become a PMA, they created what we now call the de novo pathway. So within class 2, there's a traditional 510K. You simply compare to something else. Thyroid hormone, I'm selling it, I compare it to Abbott, Roche, whoever. And I show that my test gets the same results. But I don't have to go do a trial. I don't have to do safety and effectiveness. I just do a comparison. Now, let's skip de novo and let's go to PMA. There you do have to demonstrate safety and effectiveness. And the major difference between class 3 and 2 submissions is you have to submit a manufacturing section, which is really the quality system that you submit. And you have a mandatory pre-approval manufacturing audit. That used to be very cumbersome and time-consuming. FDA has actually done a laudable job in shortening up the time frame and working with manufacturers to make that a more reasonable process. They do it in parallel with their review of your test. There's modular PMA, so you can submit this early and get this going even before your clinical study is done. So they've helped us out quite a bit. That's the major difference. There are other minor differences, but this is the major difference between a class 2 and a class 3. So now let's go to de novo. Moderate risk, but it's a new test, and there's no predicate device. I can't compare it to anything else. But I don't want to do a PMA, and FDA doesn't want to force me. They don't like them any more than we do, is my impression. I don't work at FDA, but that's certainly my impression. They're no fonder of giant submissions than we are of preparing them. So they offer this. It is class 2. You still demonstrate safety and effectiveness for a new-to-the-world test. And my rule of thumb is if it's a new-to-the-world test, FDA's never seen it before, and you're submitting it as a de novo, your burden clinically is the same as if it was a PMA. It's the same thing. You don't get a break in the sample size or whatever because it's a class 2. And if you get that kind of advice, you're probably getting bad advice, I think. It's the same thing. The big difference is you don't have to go through this. And as I said, risk-based, approximately, we can probably all argue with FDA's decisions on what is high risk and what's moderate risk, and we'll probably never all agree, but they try to do this on a risk basis. What does FDA want? I get that question a lot. And my first answer, and I'll tell you exactly what I counsel clients with, is there's no formula, there's no simple thing. FDA doesn't say, if you do the following 10 things, you're in, you're good. It doesn't work like that. Every test is different, and we have to tailor it to that. So what they want, good science and good medicine. Design a good study. Have the right patient population. Have an a priori statistical analysis plan. Statistically justified sample size. do the right things. And if your test is compelling, with compelling data, they will help you to get it through. You can write your PMA on a yellow tablet in pencil, and you're going to be fine. You know, your beautiful colors in the header and footer are not going to put a bad test over the top, and they're not going to harm a good test. FDA has said, I've heard them say, we don't care about pretty. Just give us the data. We need to look at the data. Do good science, do good medicine. The regulations are secondary. I know the regulatory folks might have a heart attack, but there are regulations and there are important formatting and things you have to do. But that's not the most important. I'm not going to go through this in any great detail. There is a pre-submission process. You're probably well aware. You should use it. There are some exceptions where maybe it's not appropriate, but in most cases, talk to FDA and go early and get their advice. It is valuable. It really is. I've done it many, many times. Laboratory developed tests, I'm not going to really describe that. They're kind of yesterday's news, I think, at this point. It's a common feature. The one thing I'll say is laboratory developed tests are illegal, ethical, moral. If you choose to offer a test as a laboratory developed test, as many companies do, including LabCorp, absolutely nothing wrong with that decision. Will they be regulated in the future? Nobody knows. Nobody knows. Right now, they're not regulated. They don't have to go through FDA, and that's a choice you can make. The one thing I would say, if you're not real familiar with our industry, standardizing a test over long periods of time is a huge challenge. And when you take on an LDT, you become a manufacturer. And unless you're trained as a manufacturer in how to keep that test giving the same result today, tomorrow, 3 years, 5 years, 10 years from now, you've taken on a big challenge, and it's not trivial. So that's the one thing that I think laboratories often underestimate. Can you be a companion diagnostic as an LDT? Obviously, yes, because 2 of those tests that have been approved recently are, in fact, LDTs. Let's go through this. I just wanted to, again, introduce these 3 tests because I think they're very important in terms of our panel discussion coming up. We're going to focus on these. One thing I want to point out is that FDA has opened several new doors with these tests, important doors, and it's changed the way, certainly the way I think about NGS testing and about multiplex testing. But one of the things that's interesting to me is that, of course, one of these tests was approved as a Class 2 de novo, the other 2 as Class 3 PMAs. In both cases, these tests, the PMAs, they point to specific therapies. So they have both tests results that say, given this result, the patient may or may not respond to a specific treatment. And then they have what are called the Table 2. We've got Table 1 and we've got Table 2 in the intended use. And there are other test results that are not of uncertain significance. But in the Memorial, they don't point to anything. They do 468, whatever it was, genes, and they simply tell you some of these genes are of probable clinical significance and some of these genes are probably not. I've forgotten the exact term. Probably not clinically significant. But the really important difference here is they had a huge database, 10,000 patient database. And to develop that database and to maintain it over time is a big deal. That involves change control, and that involves things like what happens when a variant of insignificant value suddenly becomes significant. Who's going to notify the patient? All those things become really important. So this database issue is not trivial, and as I can't think of how to do a Class 2 de novo submission not pointing to specific therapies, but simply saying probable significance, not probable significance. How do you do that without this database? I don't know how you would do that. So I think that's a big difference between the Class 2 route and the Class 3 PMA. And we can talk in more detail about these things during our panel discussion.
In fact,
so let's stop there.
Yeah, we're going to forego the questions because we're going to get into this big panel discussion.
That sounds perfectly fine.
Certainly there's plenty to speak among yourselves, but also maybe a couple of questions from the audience. So why don't you, when you're ready.
So, yeah, so if we could ask the members of the regulatory panel to come up and join us.